Hello from me. I would like to start by thanking Astrid Benne and Alexis Vasilokopoulos for inviting me to be part of this great series of seminars. And of course, all of you for attending this seminar today. And I will be talking about the work I've been doing in the past 4 years, in which years I focused on the relationships among the animal phyla. That's the first divergences, the first splits in the animal phylogeny. And these relationships back then are ancient. They have occurred more than 500 million years ago, and they are also troublesome. And by that I mean that even though we have a great amount of genomic resources to address these phylogenies, and they have been addressed multiple times by multiple researchers, they remain difficult to reconstruct, and we have not reached a consensus as a community yet. So if we look a bit closer in these relationships, and to this it helps to look at such a tree that was published in 2015, and it is based on the consensus of what we understood based on bibliography about the animal relationships. So it is a little bit out of date, but it still quite well reflects what we know and what we don't know. And we all agree on some of the relationships, like, for example, the monophily of the protostomes or the monophily of clades of protostomes, like the Ecdysozoa and the Lophotrochozoa, or even smaller clades like the chordates. But there are other parts of the tree that, even though they have been studied, we still have too many hypotheses, and we are not sure of those relationships. And the great example of this sort are the relationships within Lophotrochozoa. So there have been a lot and very different phylogenies published for this clade over the years. And there are other parts of the tree that have been less studied, so we don't know as much, but mostly because we haven't had enough material to draw phylogenies for these parts. And then there are other parts that are particularly puzzling because they have been the focus of multiple research groups with large phylogenomic datasets, and they have been debating among usually two topologies. And such relationships, highly debated relationships are on Xenambulacraria. That's a group of worm-like creatures that we are not sure if it's a sister to the rest of the Bilateria or whether it's sister to Ambulacraria. And then we have Ctenophora, the comb jellies that have been that are notoriously known for their debate of whether they are the first or the second split in the animal phylogeny. So what is the source of the challenge? The source of the challenge for phylogenetics in general and animal phylogenetics in particular can be somehow summarized in this very simple, simplified cartoon. So the shorter the time between the speciation events, the harder it is to reconstruct the order of these speciation events. And then the longer it has been since such a cluster of speciation events, it becomes even harder to reconstruct this correctly. So both of these apply in the animal, in the divergence of the animal phyla. It happened very fast and very deep in the past. And an intuitive understanding of why this is a challenge is the fact that when we have very little time in the ancestor that later split into species, it means that there is very little time to accumulate mutations that would be then inherited, and we would hope to find this information in the contemporary genomes that would be evidence of their relatedness. And not only we have limited phylogenetic signal, but we also have, if we have a lot of time since such an event has happened, then there is a lot of time to start accumulating mutations that might start overwriting the previous information. And therefore this limited, the limited mutations that had happened during the speciation events might be masked by subsequent mutations. And it's not just that, but when lineages start evolving independently for a long time, then we might start observing heterogeneous processes of sequence evolution. So it might be that some of the lineages might evolve faster than the others, or they might have a bias in their nucleotide or amino acid composition. And these both are known to cause artifacts in phylogenetic inference. And it's not only between lineages, but also parts of the genome are evolving heterogeneously. Like, for example, we might have parts of the genome that allow all nucleotides or all amino acids to be present, to be substituted with all different states, while there might be other parts that are restricted to only a subset of the nucleotides. And if we fail to accommodate this type of heterogeneities, it has been shown in different studies that it might end up in getting artifactual relationships, among which are very well known as the long branch attraction artifact. So what is, I'm sure that everybody here might have heard this before and know what is happening, but I will just very briefly and simply explain that what is happening in this case is that if you have two lineages evolving faster or being very divergent, like for example, the out group, they start accumulating a lot of mutations. And just because they start accumulating a lot of mutations, there is a greater chance that some of the mutations will be at random, similar to another fast evolving lineage. And if the information we have about the actual relatedness is very similar, this random similarity might mislead our phylogeny into clustering such lineages together. So this is the attraction of the long branches. And if we go back now and think about the animal phylogeny in this context, what might and has been discussed might be the problem is that some of the lineages, some of the phyla or clades evolve much faster than the rest, and therefore their placement might be difficult in the general phylogeny of the animals. And this is what I have worked on. So I looked into different very long branches that have been debated. And the way I saw this problem is as a difficulty in actually placing in the tree of life these faster evolving lineages. So one of them is the ctenophores, the comb jellies, and the xylomorpha, as I mentioned earlier. And then another greater clade that I have collapsed now in a single lineage is the Deuterostomes, which itself includes a lot of long branches within it. Like, for example, the flatworms or the nematodes and so on, that makes it a very long clade that can be also attracted to the outgroup. So let's start by looking into more detail of one of them, the Ctenophora, and how we dealt with this. So there are two debated phylogenies. One hypothesis assumes that it is the second split in the animal phylogeny with Porifera, which is a much simpler phylum of organisms, that's the sponges, comes first. So this hypothesis is in line with morphological understanding of animal evolution, and it has been supported by a lot of phylogenomic studies as well. But then in 2008, I think it was the first time when Dan and his colleagues first suggested that the ctenophores might actually be the first split in the animal phylogeny, and Porifera being the second. And since then, there have been more studies that do support this topology as well. So why do we care? What difference does it make? Well, the difference is how we interpret the phenotypic evolution of animals. So for example, by placing Ctenophora as the second split, what we assume is that complex characters like the origin of the nervous system or of the muscle system originated once, and it was not present in the ancestor of animals, and which is also reflected by the fact that we don't have these systems in the sponges. Of course, this is not necessarily as simplistic as I presented here, because I think there is still a debate of whether ctenophore nervous system is homologous to the rest of the animal, but it does form a very different hypothesis of how we would study this. On the contrary, if we have Ctenophora-first instead of Porifera, which is a much more complex organism compared to the sponges, then we assume at the same time that either this complex systems evolved twice, both the muscle and the nervous system evolved in two different lineages, and were probably absent from the ancestor, or they were present in the ancestor. And so we had a very, very complex ancestor of animals, and it was secondarily lost in Porifera. So as I mentioned earlier, the way I saw this problem is as a potential problem to place a long branch, that's the ctenophores, and a possible explanation is that it might be a long branch attraction, presenting the Ctenophora-first as an artifact actually rather than a true topology. And to try to find out whether that is true, we worked on two possible sources of this artifact. One was if we were not using the correct model, the substitution model to reconstruct this phylogeny, and then what is the effect of different data that are used for this phylogenetic inference. And to actually prove whether this is the case or not, we couldn't just use empirical data, because with empirical data we have the disadvantage of not knowing the truth, so we can forever be arguing which is the best phylogeny. Instead, what we did is that we used a simulation. So we used the empirical data to first learn about the different heterogeneities in the dataset, like, for example, what is the different rate of evolution among the different lineages, and what are the heterogeneities across the alignment. So is there a different composition of states across the different sites, and the different characteristics in terms of gappiness or missing sites or taxa in different data sets. So using this information, we then simulated sequences that mimic very well this characteristic in the empirical data. So they might not be exactly as the empirical data, but they mimic the empirical data, at least in the components that we would like to test with the methods. And they gave us this advantage of, in this case, knowing the truth. So we would simulate sequences under a specific topology that we could treat as the ground truth. So having now this information, we could use different methods and models to test and know whether they give us the correct answer or whether they give us a predictable error. And having done this procedure, we could then use these methods and models to either infer the relationships or interpret what we observe in the empirical data. And this is exactly what we did. We used the two topologies, the Ctenophora-first and the Porifera-first, as the ground truth, and we simulate data using parameters estimated by empirical data. And let's start seeing some of the results. So when we simulated data under the Ctenophora-first hypothesis, and we inferred the relationships using the correct model, which was a site heterogeneous model that fitted the empirical data best. So we simulated under this, and we inferred the relationships with this. Then we got always the correct answer. So this tells us, this is of course, a very happy results, because we know that if we use the correct model, we get the correct topology. But the same was true when we used the wrong model. So when we use a simpler model, a site homogeneous model that assumed that all sites evolve under the same exact rules, then we also got the correct result. And then when we looked into different qualities of data to do this phylogeny, either we used high quality data or low quality data. And by quality of data, we defined it as the type of data that can reconstruct other parts of the phylogeny that we are more certain of correctly. So regardless of what quality the data was, we still got 100% of our simulated data to give the correct topology. So overall, what this tells us is that this phylogeny is rather trivial. So regardless of the data or of the model we use, we expect to get the correct result. And then we did the same thing with the Porifera-first hypothesis, but this time we got slightly different and a bit more interesting results. So when we inferred the relationships under the correct model, we got, in most of the cases, the correct results, but in some of the cases, we got an error. And impressively, this error was not random or variable, but it was, in fact, the Ctenophora-first hypothesis. And when we used the wrong model, then we had almost in all cases a wrong topology inferred. And in all cases, this error was again the Ctenophora-first hypothesis. And then when we were using the correct model, but different qualities of data, under the high quality of data, we got the correct result in most of the cases and the error in some. But when we use low quality data, even though we're using the correct model, we got in a substantial proportion of the simulations, the error of the Ctenophora-first. So this now series of results tell us that this is a very fragile phylogeny, that if we hope to reconstruct it correctly, we need to be very careful, both with the model selection, but also with the data quality that we are using. And also this draws the picture that we observe in the empirical data, in the studies that have been published on this phylogeny. And it tells us that if the if the Porifera-first is the correct topology, then we would expect to observe as a long branch attraction artifact the Ctenophora-first hypothesis, while the other way around is not true. So this, being the truth, could not explain what we observe in the empirical data. And the same exact results we got for the placement of Xenambulacraria and Nephrozoa. So the placement of Xenosilomorpha, which is either sister to Ambulacraria or sister to the Nephrozoa. And yeah, so the result was that if the truth is the Xenambulacraria hypothesis, we would expect to observe the Nephrozoa as a long branch attraction artifact under various conditions like model misspecification, low quality data, but also when we involved a lot of long branches in the topology. But we would never observe the other way around, which again suggests that the Xenambulacraria is more probable hypothesis to explain what we observe in the empirical data rather than the Nephrozoa. So the question that one might start wondering is, why is this happening? So part of the answer could be found in the branch lengths. So what this plot shows us here is the difference in branch lengths as estimated by the site heterogeneous model, that's the blue, and the site homogeneous model, that's the orange. So both for the terminal branch lengths that are shown in this graph and the internal branches that are shown in this graph, the site heterogeneous model estimated longer branches than the site homogeneous model. And what does longer branch lengths mean? It means that it has a greater expectation of change. So the two models have a very different expectation of the change in the genomes of this phylogeny. And if we have a higher expectation of change, like the site heterogeneous model, this at the same time means that we give a higher likelihood of potential home places or random similarities. And this is the reason that site heterogeneous models are more resilient to long branch attraction artifacts, because the random similarities are not as unexpected as in the site homogeneous models. But of course, seeing this, one may wonder whether one of the models is right, or could it be that the homogeneous model is correct in estimating these branch lengths, because this is the estimation on the empirical branch lengths. So to answer that, we did another set of simulations, and in these simulations now we either assumed in the simulation site homogeneous evolution or site heterogeneous evolution. So when the sequences were evolving under site homogeneity, then both models were estimating both the terminal and the internal branch lengths similarly accurate and quite close to the truth that is shown here with this little dash. But when the sequences were evolving under site heterogeneity, the picture very much looks like what we observed with the empirical data, and that is that the site homogeneous model underestimates the branch lengths compared to the site heterogeneous model that is much closer to the truth. So we can confidently say, by seeing this result, that the site homogeneous model severely underestimates the genetic distances in such a data set. And then when we try to explain what's the difference between the quality of the data, we also saw something interesting in the branch lengths. So when we are looking into the high quality data compared to the low quality data, what we see within almost all terminal branch lengths, with one exception, is that the branches, now these are all estimated, with a correct model, the site heterogeneous model. So the branch lengths are, the terminal ones are overall shorter for the high quality data compared to the low quality data. But when we look into the internal branch lengths, we have the exact opposite. So we have higher, longer internal branch lengths with the high quality data, and shorter internal branch lengths with the low quality data. So this is like, it tells us that there is more information in the high quality data for the bifurcating events, and the terminal branch lengths are shorter. So this is the ideal situation if one would try to select data to avoid long branch attraction artifacts. And it kind of explains why under the high quality data and the correct model, we get the highest success rate in reconstructing the phylogenies, the difficult phylogenies like the Porifera-first and the Xenambulacraria hypothesis. And then after we went through these two cases, we were wondering whether a similar explanation may be the reason for the Deuterostome paraphily that has been published in some of the phylogenomic studies recently. So before I just go into this, I would just like to briefly mention that in Bilateria, that's the greatest portion of the animal phyla. We have a distinction into two big groups. That's the Deuterostomes and the Protostomes. And the difference between these two, the reason they have been clustered into these two groups, is the fate of the blastopore during the development of the animal, and whether it becomes the anus or the mouth. So when it becomes the mouth, it means that they are protostomes, but when it becomes the anus, it means that they are deuterostomes. And these two clades have been, equally and well trusted throughout the many years of zoology, like in the last 100 years, as equally robust clades. But in recent phylogenies, we have seen alternative solutions and alternative explanations of the relationships among these two great clades. So this here is the monophily of the Deuterostomes, but in a couple of recent publications, we've seen an alternative set of relationships with Deuterostomes being not being monophyletics because chordates is sister to protostomes. So we wanted to know whether it could be that the fact that the protostomes is a very long branch, or it includes many fast-evolving phyla, it might form. It might be seen as forming a very long clade that is attracted to the outgroup. So if this clade you imagine being attracted to the outgroup, then the chordates and the Xenambulacraria will be left to look as if they are monophyletic. So this was our working hypothesis. And to investigate this, we took 5 recently published large-scale phylogenomic datasets that were targeting these relationships. And we assumed 3 hypotheses. One was the as possible topologies. One was the monophily of Deuterostomes. One was the one I just explained. And then the alternative of Xenambulacraria being sister to protostomes. So this hasn't been published in any study, but we have occasionally seen it on gene trees, and therefore we assumed that it's fair to include it in our hypothesis. So could it be a long branch attraction artifact? One of the elements that we looked into using these 5 data sets was the length of the involved group. So the groups now are the Xenambulacraria, the chordates, and then we treated protostomes as a one clade and outgroups as another clade. So if it's a long branch attraction, we would expect protostomes and outgroups to be the longest branches. And this is true. So the protostomes and the outgroups are much longer than the chordates and the Xenambulacraria themselves. And this is with the original sampling in the studies in the corresponding studies. And even more interesting is the fact that in the studies that supported the Deuterostome monophyly, they had the longest, it's these 3. So this dark gray here, the orange and the gray, they supported the Deuterostome monophily, and they are the ones that have estimated the longest outgroup and protostome branches. So it makes sense that in this case, if it is a long branch attraction artifact, that we would observe it in these cases. And also when we looked into one of these data sets, and in particular the Laumer data set, and we wanted to see whether the internal branch that supports the deuterostome, so this one, is how is it supported when we use better fitting models. So this is the less fitting model, that is a site homogeneous model, and this is the best fitting model, that is a site heterogeneous model. So what we observe is that when we were going from the less to the most fitting model, the support for the deuterostome was falling. So the branch was estimated as smaller and smaller, while we had the exact opposite situation for the branch leading to the protostomes and the branch leading to the outgroup. So these well-supported splits were improving when we were using a better fitting model, but this split, that is the ambiguous one, was actually decreasing when we were using a best fitting model. And then to put it in an actual test, we did a set of simulations as the one I showed you earlier, and I will just explain what we observed. So our guide trees were the 3 hypotheses I mentioned, the monophily of deuterostomes and the 2 alternative placements of the 2 deuterostome clades with respect to the protostomes. And so when we were using the right model to infer these relationships, we got the correct topology 100% in all cases. So if this was the truth, and we used the right model, we got it in all simulations, and the same for the other 2 hypotheses. But when we misspecified the model, so we're using a simpler model than it was in reality, then in this case, we did get it right. So this was an easy phylogeny to recover. But in the other two cases, we didn't always get it right, but we got a portion of the simulations giving an alternative topology, which again was not an error. It was not a random error, but it was actually the Deuterostome monophily. So this tells us that if either of this is correct, then we would expect under model misspecification to observe the Deuterostome monophily as an error. And then we experimented by including or excluding the long branches that were sampled in this data set. So when we excluded all the long branches, then even under the wrong model, we always got the phylogeny right. So the long branches were actually exacerbating the errors. And when we excluded short branches, so in essence, we tried to do the phylogeny only included the fastest evolving taxa, then we got it always wrong. So if this was the correct or this was the correct topology, under when we had long branches and the wrong model, we would always get the monophily of Deuterostomes. So in other words, and what we can see in this cartoon here is that when these are the three topologies that we were seeing before, so this is the monophily of the Deuterostomes and these are the two alternatives, we would always get it right if we had the correct model and if we didn't include long branches in our phylogeny. But if we use the wrong model and when we had long branches in our phylogeny, then under these two hypotheses, there was a great probability that we would observe the monophily of the Deuterostomes as an error. But in this case, in contrast to the two previous ones, we are not certain which of the two is the correct topology because both of them can lead to the same error that we observe in empirical data. So could we say in all these three cases that this is it and we can assume the Deuterostome paraphily, whatever that is, or the Xenambulacraria hypothesis or the Porifera-first hypothesis? I would argue that given our current data, that these are the most probable explanations and relationships of the animal phyla, but there are still a lot of biases that we have not taken into account and might change how we perceive these relationships further. So for example, what I talked about in the introduction is that there is a possibility of compositional bias between lineages. So we have this GC that is commonly discussed, resemblance between taxa or amino acid resemblance between taxa. Then phylogenies will be artifactual in the sense that if we don't accommodate these heterogeneities, then we will tend to observe clustering of similarly, compositionally similar lineages. And then what hasn't been, so this has been discussed in the bibliography, but we haven't had yet a good way of actually addressing it besides removing data and that are problematic and hoping that we alleviate this type of problems, but it still hasn't been addressed effectively. But something that has been discussed even less and has been treated as a non-existing kind of problem is the fact that if the branches, the time between speciation events has been so short that there can be different types of problems that haven't been really addressed in deep phylogenies. And one of them is that there can be incomplete lineage sorting, and that happens when the time between speciation events is so short that the coalescence for some genes is not in the immediate ancestor of two species, but in an ancestor before. And so in this case, we would have a natural and true heterogeneity of genealogies between gene trees, and therefore concatenating them by, as we normally do in animal phylogenomics, has a great possibility of errors. And then what has been even less thought about is the possibility that if we had such short time intervals, then we cannot exclude the possibility of potential hybridization between the ancestral species, because they would be relatively still related. So these are very difficult things to model, especially if we think about all the sequence heterogeneity that we discussed and still to be explored. But it's definitely a thing to have in mind, and not to be surprised if we start working on this stuff and these relationships start being reshaping based on new methods. So just to summarize what I have said in just 4 major points is that model misspecification can explain the conflicting signal in ancient animal relationships. And the placement of Xenocoelomorpha, Ctenophora, and Deuterostomes can be affected and can be the result of long branch attraction artifact in some of the cases. And it does need careful selection of both data and models if we hope to reconstruct these relationships correctly. And what I hope I have convinced you, if not for the placement of these lineages, is the fact that using simulations along with empirical data greatly help us in identifying the optimal approaches, as well as interpret what we observe when using empirical data. But having this all in mind, we should also have in mind that several processes that are relevant to ancient traditions have still not been explored, and therefore we cannot yet be confident of the relationships as we observe them in any of these phylogenies. And with that, I would like to thank you all for listening, as well as the people that have helped me and how we have collaborated in this work. That was mainly Max Telford in UCL, and Paschalis Natsidis, part of this has been part of his PhD project, as well as with Ziheng Yang and Thomas Flory, and several other people that have been part of the collaboration, and UCL and BBSRC for funding me.